Welcome to our deep dive, everyone. Today, we're going deep into the world of antibodies,
Oh, yeah.
these microscopic defenders that are so essential to our immune systems. And we're going to unlock some of their secrets with help from this in-depth article from Assay Genie
Right.
called Comprehensive Analysis of Antibody Structure and Function.
Yeah, this is a good one.
So get ready to discover how antibodies are built, how they recognize a huge variety of threats, and the surprising ways they actually neutralize those threats.
It's true. Antibodies are often described as Y-shaped proteins,
Right.
which is accurate,
Yeah.
but there's so much more to their structure that makes them so effective.
You probably already picture antibodies as these Y-shaped proteins,
Right.
and you'd be right. But it's fascinating how this seemingly simple structure makes them so powerful and versatile. So let's break the structure down a bit further.
Absolutely. Each antibody is made up of 4 chains of amino acids,
Okay.
2 heavy chains and 2 light chains. Think of them as building blocks that form this Y shape.
Oh.
And it's this specific arrangement that gives antibodies both stability and flexibility, which are essential for their function.
So it's like a grappling hook.
Yeah.
Perfectly designed to latch onto its target.
That's a great analogy. The tips of the Y form what we call the variable regions.
Okay.
These regions are incredibly diverse, allowing each antibody to bind to a specific target, known as an antigen.
Okay, so the variable region is like a custom-made lock
Yeah.
that fits a specific key, the antigen.
Exactly.
But what blows my mind is the sheer number of different antibodies our bodies can create. How is that even possible?
It's a truly remarkable process called VDJ recombination. Imagine you have a deck of cards,
Okay.
but instead of suits and numbers, each card represents a gene segment that codes for a portion of the antibody's variable region. During B-cell development, these gene segments get shuffled and dealt out in random combinations.
So each antibody is like a winning hand in a high-stakes poker game
Yeah.
against invading pathogens. What are the chances of getting the same hand twice? Practically zero.
Exactly.
That's incredible.
This random shuffling of gene segments allows for the generation of billions of unique antibody combinations. It's like having an army with billions of soldiers, each trained to recognize a different enemy.
Wow. That's an incredible level of preparedness.
Get me out.
It doesn't stop there, does it?
Yeah.
This article also mentions something called somatic hypermutation,
Uh-huh.
where the antibodies actually evolve over time.
You're right. Even after those B cells have shuffled their gene segments,
Right.
they can further refine their antibodies through somatic hypermutation. It's like taking that winning hand and fine-tuning it to be even more effective.
So after an antibody encounters an antigen, its genes undergo these small mutations.
Yes.
And if those mutations make the antibody even better at binding to its target, that B cell gets a signal to multiply and produce more of those highly specific antibodies.
Precisely. It's like natural selection happening at a microscopic level, ensuring that the most effective antibodies are the ones that proliferate. And this process allows our immune system to adapt and become more efficient over time, especially when encountering the same pathogen again.
This intricate structure and incredible diversity are just the beginning. What's fascinating is that not all antibodies are created equal.
Absolutely. We have different antibody classes, also known as isotypes, each with its own specialized function.
It's like a diverse team of specialists within our immune system, each playing a critical role in defending us from harm.
Exactly. Think of it as a well-coordinated defense force. We have IgG, the most abundant type found in our blood and tissues. It's the workhorse of the antibody world, able to neutralize toxins, viruses, and bacteria.
So IgG is like the frontline soldiers always patrolling and ready to fight off invaders wherever they might appear.
Then we have IgA, which is found primarily in mucosal areas like our respiratory and digestive tracts. These are the antibodies that stand guard at the entry points, preventing pathogens from getting a foothold in the first place.
IgA is like the border patrol keeping those unwanted visitors from ever crossing into our territory.
Now, IgM is the first responder appearing early in an infection. Think of it like the paramedics of the immune system arriving quickly to contain the damage and alert other immune cells to the threat.
So IgM is like the first wave of defense, buying time for the rest of the immune system to mobilize. What about IgE? I know that one is involved in allergies.
Yes, IgE is often associated with allergic responses, but it also plays a critical role in fighting off parasites. These antibodies bind to the surface of parasites and signal other immune cells to attack.
So while IgE can sometimes be a nuisance with allergies, it's also our defender against those larger, more complex invaders.
Exactly. And finally, there's IgD, the most enigmatic of the antibody classes. It's found on the surface of B cells, but its exact function is still being investigated.
IgD is like the mystery guest of the antibody world, still revealing its secrets to researchers. This is all so fascinating. It's amazing how each antibody isotype is specifically designed to tackle a different type of threat.
It really is. It's like having a specialized team for every possible scenario, ready to defend us from any angle.
It really highlights the complexity and efficiency of our immune system.
Hmm.
But we were discussing those remarkable effector functions, the actions antibodies take once they've bound to their targets.
Right.
It's not just about recognition, it's about taking action.
Absolutely. Antibodies are far from passive players.
Mm.
Once they've latched onto their target, they can trigger a variety of responses that ultimately lead to the neutralization or elimination of the threat.
It's like they're sending out a signal flare, alerting the rest of the immune system to the presence of an intruder and directing the appropriate response.
That's a great way to put it. One of the key effector functions is neutralization, where antibodies physically block a pathogen from binding to and infecting our cells.
So the antibody is putting itself in the line of fire, acting as a shield to protect our cells from harm.
Exactly.
Huh.
For example, imagine a virus trying to attach to a cell receptor in order to infect it.
Okay.
A neutralizing antibody can bind to the virus, covering up its binding sites and preventing it from attaching to the cell.
It's like the antibody is wearing a disguise, mimicking the cell's receptor and tricking the virus into binding to it instead.
That's a great analogy.
Ah.
And by binding to the virus, the antibody effectively neutralizes its ability to cause infection.
Yeah.
It's a very direct and effective way of preventing the spread of a pathogen.
That's incredible. It's amazing how such a small molecule can have such a powerful impact. But what about those situations where a pathogen has already managed to enter a cell? Can antibodies still help in those cases?
Absolutely. That's where other effector functions come into play, like opsonization.
We briefly touched upon opsonization earlier. It involves antibodies tagging invaders for destruction, right?
Exactly.
So-
Remember the FC region, the tail end of the antibody's Y shape?
Yes.
That region plays a crucial role in opsonization.
Right. The FSC region acts as a communication hub, interacting with other parts of the immune system.
Precisely. In the case of opsonization, the FEC region of an antibody can bind to receptors on phagocytes, which are specialized immune cells that engulf and destroy pathogens.
So the antibody is holding on to the pathogen with one hand and waving to a phagocyte with the other, saying, "Come and get it."
Exactly. The antibody acts as a bridge, bringing the pathogen and the phagocyte together. This tagging by antibodies makes the pathogen much more appealing to phagocytes, increasing the efficiency with which they are destroyed.
So the pathogen gets tagged and then gobbled up by the phagocyte. That's quite a visual. But what about complement activation? That one sounded a bit more explosive.
It is. The complement system is a complex cascade of proteins that can directly destroy pathogens by punching holes in their membranes. It's a powerful weapon in the immune system's arsenal.
So how do antibodies trigger this cascade of destruction? Is it through that all-important MFSA region again?
You got it. When antibodies bind to a pathogen, their FC regions can change shape, exposing a binding site for the first component of the complement cascade.
It's like a domino effect. One thing triggers the next, ultimately leading to the destruction of the pathogen.
That's an excellent analogy. It's a very tightly regulated process. But when activated, it can rapidly and efficiently eliminate pathogens.
We've covered neutralization, where antibodies act as blockers, opsonization, where they tag invaders for destruction, and complement activation, where they trigger a cascade of proteins that directly destroy pathogens. Are there any other tricks up their microscopic sleeves?
There's one more key effector function we need to discuss: antibody-dependent cellular cytotoxicity, or ADCC.
Ah, yes, ADCC. The one where antibodies call in the big guns, natural killer cells, to eliminate infected cells.
Precisely. Natural killer cells, or NK cells, are a type of white blood cell that can directly kill target cells, and antibodies act as guides, leading them to their targets.
So it's like the antibodies are placing a hit on infected cells, marking them to destruction by the NK cells.
That's a very apt description. The antibody binds to a specific antigen on the surface of the infected cell, and then its FC region interacts with a receptor on the NK cell.
It's another example of the FC region acting as a communication hub, sending out signals and directing the immune response.
Exactly. This interaction activates the NK cell, triggering it to release cytotoxic granules that destroy the infected cell.
It's amazing how antibodies can orchestrate such a precise and targeted attack. They're like the ultimate puppet masters, controlling the immune system's every move.
It's a testament to the intricate and elegant design of our immune system. And it highlights a key takeaway from this article. Antibodies don't work alone. They're constantly collaborating with other parts of the immune system to ensure our protection.
It's like they're the conductors of the immune orchestra, bringing all the different instruments together to create a harmonious defense symphony. This is incredible. We've uncovered their intricate structure, the incredible diversity, and the remarkable effector functions they use to fight off invaders, from neutralizing pathogens to tagging them for destruction, and even calling in reinforcements like natural killer cells. It's clear that antibodies are way more than just simple sticky molecules.
They really are.
Mm.
And this deep dive has only scratched the surface of this field. There's still so much we don't know about antibodies, and scientists are constantly making new discoveries.
That's what makes this field so exciting. This article has been incredible, but it's sparked a whole new set of questions for me.
Oh yeah, like what?
Like, what are the limitations of antibodies? Are there situations where they might not be effective? Or what about new antibody-based therapies? What are some of the challenges and opportunities in that area?
Those are great questions, and they're really at the forefront of research right now. For example, one area of intense focus is on developing antibodies that can target cancer cells.
So using antibodies as a magic bullet to destroy cancer cells without harming healthy cells, that sounds pretty promising.
It has huge potential. And there are already several antibody-based cancer therapies available with more in development, but there's still some challenges. For example, cancer cells are known for evolving and evading the immune system.
So it becomes a sort of arms race with scientists constantly trying to develop new antibodies that can keep up with the cancer cells.
Exactly. It's a fascinating and challenging field, but the potential benefits for patients are huge.
Another area I'm curious about is the role of antibodies in autoimmune diseases, where the immune system attacks the body's own tissues. Can antibodies be involved in that?
Absolutely. In autoimmune diseases, the immune system goes rogue and produces antibodies that target the body's own cells and tissues. This can lead to a ton of different symptoms and complications depending on which tissues are being targeted.
So the antibodies are actually contributing to the problem instead of solving it.
Exactly. And understanding the role of antibodies in autoimmune diseases is critical for developing effective treatments.
It's clear that antibodies are super powerful molecules with the potential to both harm and heal. Seems like a delicate balance.
Yeah.
And scientists are still unraveling all the complexities of this system.
That's true. But with every new discovery, we get a deeper understanding of how antibodies work and how we can use their power for good.
This deep dive has been an incredible journey into the world of antibodies. We've explored their structure, their diversity, and their ability to orchestrate a complex immune response that keeps us healthy. But our exploration has just begun.
It really has. And as we continue to learn about antibodies, we can expect more groundbreaking discoveries that will transform how we approach disease and develop new therapies.
So as we wrap up this deep dive, I encourage you, our listener, to keep those questions in mind. Don't let your curiosity end here. Dive deeper into the world of antibodies, explore the latest research, and see where your journey takes you. The world of science is full of wonders just waiting to be uncovered.